go to the BMS 300 homepage and find the site where it associates the first letter of your last name with the room you're supposed to be in today. And we'll meet together again at 5 at all those various rooms, and we'll have that exam. So what we're going to do today is we're going to start a new topic. We're going to start into the cardiovascular and blood and immune section. And so the first discussion here is basically going to be about the mammalian heart, its structure and function. By the end of the discussion today, we'll have gone through the chambers of the heart, their vessels and the valves, and the blood flow through these chambers and across the valves. In order to understand why we have to have a heart, I want to spend a little bit of time just on the reasoning for why a heart is so important. If we go back in time and we look at early organisms, we'll see that as long as the organism was made of a fairly small number of cells, and that those cells were within 1 to 2 millimeters of the source of the oxygen or nutrition, they didn't really need to have a circulatory system. They really didn't need to have a heart. But once you begin to expand an organism beyond this sort of couple of cell layers or a single cell, then we have to do something about moving the nutrients and the oxygen to those cells. Sort of a rule here is that the distance of diffusion that can keep a cell alive is only about 1 to 2 millimeters. That's all the farther we can get significant diffusion of things like oxygen and nutrients or waste products. And so what we need then, once we get to see larger organisms, we need to then have some means of bringing the fluid, which used to be the ocean, to all of these cells within 1 to 2 millimeters. And that's the genesis, that's the idea of having a heart and a vascular system, a series of tubes through which we can move the fluid. I don't want to spend a whole lot of time on it, but we'll work our way through a single-celled organism, an organism such as a sponge or a jellyfish, which is only a couple of layers of tissue thick, cells thick. And then we'll work our way into a three-dimensional organism in terms of a teleost fish, a goldfish, if you will. And I want to spend a little time on this simply because it shows why we need a heart, but it shows how, at least for the teleost fish, you can have a fairly simple structure doing all the jobs the fish needs. We'll see that in the fish, there's a two-chambered heart. It has an atrium that receives deoxygenated blood, and then a ventricle that will pump that deoxygenated blood to the oxygenating organ. The oxygenating organ being the gills. We've all watched a goldfish in a tank, and we can watch the operculum move. And as the operculum moves, what's happening is that fish is pumping water across its gills. And its gills are the oxygenating organs. We'll spend a few moments here describing how this single ventricle is going to pump the blood first through the gills, and then from the gills through the rest of the vasculature. But there's only one pump in this system. One pump does the whole job, both for the oxygenating organ and also for the systemic vasculature. That's not true once we begin looking at birds or mammals. Birds or mammals have a four-chambered heart. There's some argument about whether or not birds and mammal hearts have the same evolutionary origin. But nonetheless, we ended up in both cases with two pumps, two-chambered hearts. One pump is to pump the blood to the oxygenating organ, to the lungs. The other pump is to pump the blood to the systemic vasculature, all the other organs. And these two pumps are working in series. So the blood which has been moved to the lung is going to be brought back to the left side and pumped out to the rest of the vasculature. So there'll be a circuit. Deoxygenated blood is going to enter the right side of the heart. That's the pulmonary pump. It's then going to be pumped from the right side through a series of tubes. Primarily the one we're going to talk about is the pulmonary artery. The pulmonary artery is carrying blood away from the heart and toward the lung. Then that blood is going to be oxygenated in the lungs because that pulmonary artery is going to branch and branch and branch and branch into tiny, tiny capillaries. And that's the site where oxygen will enter the blood and reoxygenate the blood. That oxygenated blood now is going to leave the pulmonary vasculature and come back to the left side of the heart. It's two pumps in series. So the right heart, the right ventricle is going to pump blood to the lungs, where it's oxygenated. That blood now is going to return to the left side of the heart. But again, it's going to look like a very similar system. It's going to come into a chamber called an atrium on the left side. And then from the atrium on the left side, it's going to move into another chamber called the ventricle on the left side. And then from the ventricle, that blood now is going to be pumped out to the systemic vasculature through the aorta. And we'll talk about all of those structures. But for now, hold on to these two major ideas. Right atrium and right ventricle is the pulmonary pump. Left atrium, left ventricle is the systemic pump. And we'll initially draw this as a kind of 4-box model, just to show how the blood is going to flow through these chambers or rooms, and how there are going to be doors or valves between the chambers, and additional doors or valves from the ventricular chambers, either right or left, and into the tubes carrying the blood away from the heart. So we'll talk about the chambers, we'll talk about the valves, and we'll see that there's a pulmonary valve at the opening of the pulmonary artery, and there's going to be an aortic valve at the opening to the aorta, and we'll walk our way through all of that. But let's start with a really simple system. Let's imagine, a couple billion years ago, 1.5 billion years ago, there was a single eukaryotic organism. It lived basically in the ocean. The only separation between the cytoplasm of this organism and the ocean was the plasma membrane. So that means that oxygen could easily diffuse in. And also nutrients could simply diffuse across that phospholipid bilayer. But then being alive generates certain byproducts. And one byproduct from, if it has mitochondria, from oxidative phosphorylation. The byproduct of oxidative phosphorylation in the mitochondria, of course, is carbon dioxide. That can easily diffuse out across the membrane. Similarly, there's another byproduct, which is ammonia. Ammonia is a byproduct of protein metabolism. Ammonia is pretty toxic, so cells don't tolerate much of it. But if you're a single-celled organism, it's not really a problem because the ammonia can simply diffuse right out. The problem doesn't get too much more dire if we imagine something like a jellyfish or a sponge, and particularly in the case of the jellyfish, there's a layer of cells on the outside. This is called the ectoderm. And then there's sort of a crest here, and it continues to the inside, and I'll put this in orange, and there's another layer of cells. This is called the gastroderm. And then there's basically the jelly in between, to give this the name of the jellyfish, which is named the mesoglia. Now, I don't want to spend too much time on jellyfish, but I do want you to see that if you ever watch a jellyfish, you notice that bell pulsates. What it's doing is it's bringing bringing ocean water in to approach the gastroderm and back out again. But the issues of simple diffusion can solve the problems for this organism as well. It's a single layer of cells, basically, on the inside, and a single layer of cells on the outside, so oxygen can easily diffuse into those cells. Nutrients can easily diffuse in. And carbon dioxide and ammonia can easily diffuse out. So simply by layering cells into a couple of layers, we still retain the advantages of a single-celled organism, and we don't really need a cardiovascular system. We don't need a pump and a series of tubes to carry the blood. But now let's think over here. I'm going to draw one of my dreadful drawings. This is meant to be a teleost. Fish. This is like a goldfish. These are all the bony fishes. True bone teleost. And I'll draw sort of a facsimile of one of these. It's a tail. There's a dorsal fin. There's a ventral fin. Then up here's a head. There's an eye. And there's a mouth. And what the fish is doing is it's bringing water in through its mouth. And there's the structure back here. We've all seen it in looking at a fish. We commonly call this the gill, but it's actually the operculum. The operculum is that bony structure that which you watch moving in and out. What the fish is doing is it's bringing water in through its mouth and forcing that water across a deeper structure down here, which will be the gills. The gills are the oxygenating organ. Well, the fish has to, in some way, deliver that oxygen to all of its tissues. So it uses a pump. But it's a pretty simple pump. If we look at the pump, there are two chambers. There's an atrium, and up here, there's a ventricle. And the way the system functions is that deoxygenated blood, deoxygenated, enters the atrium. The atrium is the receiving structure. It's a receiving chamber. So deoxygenated blood coming back from all the tissues enters the atrium, enters the ventricle. But the ventricle is muscular. And when that muscle contracts, it forces blood into a series of tubes that go to the gill. So we're taking in deoxygenated blood from the tissues into the atrium, putting it in the ventricle. Ventricle then pumps the blood to the gills. But what's interesting is two things. First is there's only one pump. There's one pump that pumps blood to the gills. And let me draw the gills here to give you some idea what the gills are doing. The gill is a pretty simple structure. It basically has a layer of epithelial cells. So we can use this as a hash. That's the epithelial cells. The epithelial cells will sit on a basal lamina, so that gives you the orientation of those cells. And out here is water, because this is the outside world. And then immediately beneath those epithelial cells will be a blood vessel. I'll try to do my best to draw this in some sort of... I'm trying to show is that blood vessel is going to be composed of a continuous layer of endothelial cells. So these are all meant to be, I won't do them all, but these are meant to be endothelial cells. And the blood, the oxygen-poor blood, flows in this direction. And as it flows in that direction, oxygen comes out of the water, diffuses out of the water. The oxygen diffuses out of the water, goes across the epithelium, goes across the extracellular space, goes across the endothelium, and into the blood, where it can be picked up by erythrocytes and carried to the rest of the vasculature. So if we go back to the upper panel, what we're showing is that this muscular ventricle is pumping the blood to the gills. But that blood in the gills then flows through the gills as we showed above, but that blood then goes out to the systemic vasculature. And what I mean by the systemic vasculature is just all of those blood vessels that are now going to feed oxygen to all of the other tissues. It doesn't matter whether it's muscle or gut or brain. So there's one pump in the system to serve the whole thing. So this very same pump that's moving blood through the oxygenating organ and getting the blood to be oxygenated will now force the blood through the tubes to the remaining tissues. Fish do just fine, but it's not a very efficient system. And oddly enough, if you look at some cephalopods, such as squids, which are really highly active, you'll find they have solved the problem. What they have done is they have generated one pump that feeds the gills, blood, and then they have another pump which takes that blood on the far side of the gills and pumps it to the rest of the vasculature. So you have these two pumps that are in series. We're going to modify that system. I mean, that was a totally different evolutionary system. But from this system of one pump, of an atrium and a ventricle, if you look at it developmentally, this structure is going to fold over on itself and it's going to generate the four-chambered heart of the mammalian heart. If you take an embryology course, it's really interesting to watch how this thing folds and ends up being a four-chambered heart. But what we're going to talk about is the idea of the four-chambered heart. A mammalian four-chambered heart. I'm going to simplify this down to basically 4 rooms. And you can imagine walking through the 4 rooms. We'll give those rooms names and we'll give the doors to all the rooms names, but I want to just walk our way through the rooms that are going to be the two sides of the heart. The first thing I have to tell you is that the convention is that when I draw this up here, it's as though you're looking at me. It's as though my heart were going to be plastered on this board. And so this is going to be right, and this is going to be left. So imagine on the board like this, and then you can go right and left. When we do that, and the convention is to use the right in blue and the left in red, I'm going to draw a box here. And I'm going to label this box as the right atrium. Term atrium comes out of the Latin. Discuss this a little bit. This is a receiving area. If you go into a really big building, so you walk into a skyscraper someplace in Denver, and you immediately walk into that building, there's this great open area, that's the atrium. It's a receiving area. And what we're going to do now is we're going to be receiving blood into this room, into this chamber. And this chamber is going to be the right atrium. You can think of it as a room. There's going to be fluid coming into that room. And the fluid is going to come in a structure here known as the vena cava. The vena cava is a single tube. It originates as a fusion of the superior and inferior vena cava. Now, So we have the inferior coming in this direction, superior coming from the head. The blood comes into this site and enters the room from the vena cava into the atrial chamber. There's another room down here. This other room is called the right ventricle. There's a door between the two rooms. There's a door right here. We give that door a name. We call it the right atrioventricular valve. So this right atrioventricular valve is the doorway between the atrium and the ventricle. We're going to see when we walk our way through this that this deoxygenated blood coming in through the vena cava is coming through the atrium, flowing through the open door and into the ventricle. Then there's one path out from the right ventricle. And this path out is the pulmonary artery. There's convention for this, and it's worthwhile holding on to. The convention says that an artery carries blood away from the heart. So we're going to be carrying blood away from the heart. In this case, it's going to be O2 poor blood. It's going to be carried away from the heart in the pulmonary artery. Pulmonary tells you it's carrying the blood to the lungs. So this is the right heart. This is the right heart that is dedicated to moving blood to the lungs. So the blood is now going to come as deoxygenated blood into the right atrium, flow across the atrioventricular valve into the right ventricle, and then there's going to be another valve here. This is going to be the pulmonary valve. The pulmonary valve is at the opening between the ventricular chamber and the tube that's going to be carrying blood away from the heart and toward the lungs. Well, I need to walk you through some of the other nomenclature because you're likely to run into it. I much prefer the term right atrioventricular valve. It tells me where it is. It tells me on the right side. It tells me it's between the atrium and the ventricles. It's the right atrioventricular valve. But commonly, you'll see this named anatomically. Commonly, you'll see it named as the tricuspid valve. And there's some meaning in this. The tricuspid valve looks a little like this. And there's another one behind the plane of the blackboard. It has 3 leaves. Tri is 3, cuspid meaning leaves, so it's 3 leaves. So the right atrioventricular valve and the tricuspid valve are the same thing. The flow of blood here is deoxygenated blood coming in from the vena cava, coming into the right atrium, across the right atrioventricular or tricuspid valve, into this room. And then it's going to leave this room and go out into the pulmonary artery and be carried to the lungs. But we said that this right heart is the pulmonary pump, but the other side is the systemic pump, and that the two pumps are arranged in series. And what we mean by that is that over here, another box, this is going to be the left atrium. You can think of this as an open room. That's fair. But there's going to be a feed of fluid into that open room. And this feed of fluid is the pulmonary vein. Now again, there's nomenclature here. When you hear the term vein, just as we saw over here with the vena cava, we were carrying blood to the heart. Here we're carrying blood to the left side of the heart. So this is a vein. But in this case, this vein is carrying O2 rich blood from the lungs. So that's the pulmonary vein. Then, just as we saw before, we'll have these two rooms. Where on the right side we had the right atrium. Here we have the left atrium. Here we have the left ventricle. And again, there's a door between the two rooms. And this we can label as the left atrioventricular valve. Then coming out of the heart is another tube. This tube is said to be the aorta. There's only one tube leaving the ventricular chamber. Now this tube is going to branch and branch and branch. Doesn't take very long after leaving before this branches into several other major vessels. We're not going to go into that. If you take an anatomy course, you'll name all of them. I just want you to see that everything leaving the heart goes out into the single tube called the aorta, and that between the chamber here is the aortic valve. The aortic valve has another name. It's also, again, an anatomical name. It's called the left semilunar valve. The pulmonary valve also has another name. The pulmonary valve over here has another name. I'm running out of room. Known as the right semilunar valve. I prefer pulmonary valve and aortic valve. It tells me where it is, but you'll sometimes run into these terms. My understanding is where this term semilunar came from is when, during the Renaissance, they began doing gross anatomy and they began looking at the heart. They looked at these valves both in the pulmonary artery and also in the aorta, and they said they looked like half moons. They called them semilunar. And I don't find it very useful, but you'll often see that nomenclature. It gets even worse for this valve. This valve, the right, put it in red, the right atrioventricular, the right atrioventricular valve has another name. It's called the bicuspid valve. So it basically has two leaves, if you look at it this way. So it's bicuspid, there are the two leaves. But it has an even more arcane name. And you'll often see this referred to as the mitral valve. Again, this is left over from the Renaissance, and it was called the mitral valve because if you turn this thing upside down, it looks a little like this. And if you've ever seen a bishop, or the Pope for that matter, who's the Bishop of Rome, all dressed up for a special occasion, where they wear these really weird hats that are composed of these two more or less triangular shaped structures, and this is called a mitre. That official hat that a bishop wears is called a mitre. So it was given the name the mitral valve because it looked like these two leaves. Well, let me show you a couple of pictures, and then we'll come back and try to put all of this into context of a proper heart. The first picture is just a pretty picture of what I told you about with the fish. It just summarizes in a pretty picture what I tried to show in my ratfish image. So this is meant to be the gill here. If you come back here, this is the atrium, and this is the ventricle. The flow of blood, of deoxygenated blood from all the organs comes into the atrium, flows into the chamber or the ventricle, but the ventricle is muscular. So it contracts, and when it contracts, it sends blood to the gills. But this very same pump that's pumping the blood to the oxygenating organ, once the blood leaves the oxygenating organ, there's no other pump. It's only a single pump. That blood then goes out to the rest of the systemic vasculature, and so this one pump has to provide blood to both the oxygenating organ and the rest of the vasculature as well. What we've just seen is we don't do that in mammals. We have two hearts in mammals, basically, one dedicated to the pulmonary circuit and one dedicated to the systemic circuit. But because of the blood flow from the right heart to the lungs, back to the left heart, those two pumps are said to be in series. This next picture is to help you think of the idea of the mitral valve. This is a picture of Rihanna, of course, and this was taken several years ago at the Met Gala, and I think the theme was spirituality or something. And so Rihanna decided to go dressed as a pope, or as a bishop. So she got a hold, I think this is actually the mitre from the Bishop of Milwaukee. But she dressed up with this, and I like it because if you turn Rihanna upside down, you can imagine the blood flowing through that mitral valve. This is the mitre. And so it's the two leaves of the bicuspid valve. It's the it's the left atrioventricular valve. So, any questions to there? Yes?
Should that be the left atrioventricular?
What did I do?
Kind of underneath the bicuspid.
This should be the left atrioventricular valve.
Where you said it? This is R right there. Yeah.
Oh, thank you. This is the tricuspid. This is going to be the right atrioventricular valve. And this, and I did misspeak, this bicuspid valve should be the left atrioventricular valve. Thanks. That's right. Didn't mean to confuse you. So the bicuspid valve's on the left. It's the left atrioventricular valve. Tricuspid's on the right. It's the right atrioventricular valve. Good. Anything else? All right. What I'm going to do is I'm going to repeat this pattern. I'm going to go from the 4-box model and try to simulate the structure of the heart. This still won't be anatomically correct, but it'll be closer than the 4-box model. And we'll begin to see not only where the valves are and where the chambers are, we'll begin to see the idea that all of these chambers are surrounded by a muscular wall. Now, it's true that the muscular wall on the ventricular side is much, much thicker than the ones for the atria. But still, there are these muscular walls. So let's make one of these sort of simple diagrams, really just doing nothing more than speaking again of what we have in the upper panel. So the structure here, this is the right atrium. So we can label this as the right atrium. This is the atrial myocardium. Now there's meaning in this term myocardium. Myo means muscle. It's the muscle of the heart. So surrounding these chambers, surrounding the opening is this wall of muscle. This is going to be the atrial myocardium. The blood flow in, as we showed above, will be through the vena cava. And down here is going to be, of course, the ventricle. So we'll show the ventricular chamber as an opening. But that ventricular chamber is surrounded by this ventricular This is a ventricular myocardium. It's a wall of muscle. Just as we showed above, there's a valve here. So the only opening between the chamber of the atrium and the chamber of the ventricle is this right atrioventricular valve. The right atrioventricular valve is also known as the tricuspid valve. The blood flow is going to be just as we showed. Comes in. We'll see later this is a low pressure. There's not very much pressure on this. Flows across those atrioventricular valves and into the ventricle. Then the output from the room, the output from the right ventricle, is this structure here, which is the pulmonary artery. And at the interface between the chamber and the opening of the tube is this valve, which is the pulmonary valve. And this valve, when it's open, will allow the blood to flow out. We can show basically a similar story for the systemic heart. So again, here's the left atrium. So this is the left atrium. It has an atrial myocardium. And it has a tube flowing into it, which of course is the pulmonary, coming from the lung, vein. Veins carry blood toward the heart, tells you this is the origin of this blood is in the lungs, being carried toward the heart as a vein. Comes into the heart again, just as we see on the right, we see on the left. Pretty similar pattern. It turns out the wall of the left ventricle is a little thicker than the right-hand side, but it's the same general pattern, where this is the myocardium. And this is the chamber. And now on the way out, there's another tube here. We call this the aorta. And again, there's an aortic valve. So the O2-rich blood, which is returned from the lungs, comes into the left atrium, flows across the left atrioventricular valve, and the left atrioventricular valve now allows blood to flow across it into the ventricles. Now, I'll mention this here, and we'll come back to it a little later. What we're going to do here is we're going to take the myocardium on the left and the myocardium on the right, and it's going to contract almost simultaneously. And when it contracts, what it's going to do is it's going to generate pressure on the fluid inside the chamber. When we generate pressure on this fluid, these floppy valves, the atrioventricular valves are really floppy, they're kind of loose. So when we increase the pressure, it snaps those two valves shut. When you're listening to a heart, that's the lub sound. Then the pressure builds for a little bit. We continue to build pressure and we continue to build pressure until the pressure in the chamber, I'll say that pressure in the chamber is greater than the pressure in the aorta. Or we could say on the right side, it's true as well, the pressure in the chamber is greater than the pressure in the pulmonary vein. When that pressure on the chamber side is greater than on the far side, in both cases, this valve opens. Now, in contrast to the atrioventricular valves, which are pretty floppy, they're pretty loose, these valves are pretty darn stiff. It takes a lot of energy to open these valves. So we're going to use the energy of the muscle contraction, increasing the pressure on the fluid in the chamber to open those valves. It's actually worth thinking about this in terms of doors. And it's worth thinking about this in terms of doors we have in this building. And I don't know how many of you have done this, but I've done this 1,000 times. And I go to the door out here and I think, I'm just going to walk out this door. And so I hit the breaker bar and nothing happens. And so I get my arms into it and nothing happens. And then finally I remember, oh yeah, there's a lot of pressure on that. And then I get my legs into it and finally the door opens. But what you have to do to open that door, because the inside of this building is a negative pressure with the outside. There's a greater air pressure outside this building than inside it. So in order to open that door, you have to put more pressure on the inside of that door than the air pressure on the far side of the door, forcing it shut. That's exactly what's happening here. We have to increase the pressure inside the chamber to force this door open in such a way that the pressure on this side is greater than on this side. The way it ends, though, is that after the blood leaves, that we'll have pressure, some residual pressure in here, and when the pressure on the aortic side is greater than the pressure on the chamber side, then that valve slams shut. And that's the dub sound of lub-dub. And you had a question and you had one behind, so let me take yours first.
My question was more about the pressure concept. You said that when, they both sides simultaneously come together. So would the pump from the ventricles cause the shut door on the tricuspid valve?
Yes.
So it would close the tricuspid valve while opening the aortic valve?
Well, we'll see there's a delay, and we'll go into this in more detail. But right now, you can imagine that when we contract the muscle, we increase the pressure. When we increase the pressure, these are floppy doors.
Yeah, those will close, correct?
And those will slam shut. Kaboom. And then, but there's a delay while the contraction increases and increases and increases, forcing more and more pressure on the fluid until the pressure in here gets so high that this stiff door out here finally opens. Now, it turns out that this valve is stiffer than this valve, and the pressures generated on the left side are greater than the pressures generated on the right side. The pressures here will be upwards of 100, 110 millimeters of mercury. This will be down around 30 or 40. We just don't need as much pressure to force the blood through the pulmonary circuit as we do the systemic circuit. But it's the same idea. You have to have a greater pressure on one side of the door than the other to swing them open. Yes? Okay, got it. Now there's another one over here someplace, I thought. Okay, not. So that's basically how the blood is flowing through the system. The next thing I want to talk about is that the chambers are lined with a single layer of endothelial cells. The endothelial cells are confined to the cardiovascular system, and we're going to run into them over and over again. But for today's discussion, we can say that if we think about these chambers are lined, they're sort of like wallpaper on these chambers. And that wallpaper is composed of these individual cells lined up one next to the other. This is an endothelium. Wherever you go in this system, whether it's in the chambers, whether it's on the surface of the valves, whether it's on the surfaces of the vessels, there's this continuous layer of endothelial cells. That's true on this side as well. So there's a continuous layer of endothelial cells that line the chambers and line the valves. I won't draw them all the way around, but also continue out into the veins. So if we follow the sequence of blood through the system, do it here, we see that the pattern is that when blood flows away from the heart, it enters the aorta. It's a single tube. Then from the aorta, it goes into arteries. These are many, many branches of the aorta. From the arteries, it goes to the arterioles. Now from the arterioles, these branch and branch, and they break down into structures, which I'm going to put in green here, called capillaries. Capillaries are in fact the exchange vessels. It's at this site where O2 leaves the blood vessel. We say capillary. And carbon dioxide diffuses in. So that's where the exchange is. If we were talking about nutrients, it'd be the same thing. If we're talking about ammonia, it'd be the same thing. That's where the exchange occurs. And these guys, the capillaries, are composed of a single layer of endothelial cells. They're exquisitely thin. It's because they're so thin, because these endothelial cells are so thin, that we can easily get diffusion across them. But back to the very first discussion, these capillaries will find their way into all of your organs, and all of your cells will be within 1 or 2 millimeters of these structures. And these are the exchange vessels. But then from the capillaries, that blood is gathered together into another set of tubes called venules. And the venules will drain into veins, and the veins will then drain into the vena cava. And from the vena cava, of course, we go back to the right side of the heart, and we start the circulation all over again. But this brings us back to the main idea that arteries carry blood away from the heart. And veins carry blood toward the heart. And that's the convention. So again, when we talk about the aorta and the arteries, is carrying blood away. But similarly over here, the pulmonary artery, even though it's carrying deoxygenated blood, is carrying blood away from the heart toward the lungs. The pulmonary vein is carrying oxygenated blood, but it's carrying that oxygenated blood from the lungs back to the heart. So it's headed toward the heart. Well, that's about as far as I got in the first one. So let's stop there. And tomorrow we'll come back to another set of cells that will be on the outside of the heart, forming the capsule in which the heart is contained.